Albert Leninger is often regarded as the father of modern biochemistry and made considerable contributions to our understanding of the electron transport chain. After studying fatty acids and their relation to the Krebs cycle in 1945, Leninger continued his studies of cellular metabolism with mitochondria in 1948. After scientists at the Rockefeller Institute had isolated mitochondria via differential centrifugation, Leninger found that the mitochondria was the only place that could break down the fatty acid chains he had previously studied. We performed centrifugation when we separated our liquid samples a few weeks ago. In just 3 years, Leninger had identified the location of nearly all major energy reactions in the cell.
By isolating mitochondria and studying it as an independent organelle, Leninger also promoted ideas surrounding compartmentalization, which scientists use when studying cells today. Then, in 1956, Leninger proved that the electron transport chain and ATP synthase were only located in the mitochondrial membrane. In 1958, Leninger developed evidence for oxidative phosphorylation, and scientists at the time accepted that ADP was converted into ATP at this stage. This idea is known as energy coupling. Leninger laid the groundwork for the electron transport chain, but Peter Mitchell expanded on it in the 1960s in his exploration of chemiosmosis and the role of a proton gradient in the ETC. Mitchell realized that the movement of ions across an electrochemical gradient could provide the energy needed to produce ATP. Ions in an area of high concentration move to an area of low concentration through active transport. Throughout the mid to late 1900s, Leninger and Mitchell were rivals, both trying to disprove the other's theories and promote their own. Either way, the two scientists made enormous contributions to the field of cellular metabolism, and their ideas are still used in studying the electron transport chain today.
In cellular respiration, the electron transport chain is a series of enzymes bound to the inner mitochondrial membrane in organic molecules.
What in the world did you just say?
Oh, sorry. Let me backtrack a little. The ETC is started by NADH and FADH2, important electron carriers produced during glycolysis and the Krebs cycle. The electrons they carry travel through a series of enzymes that act as different energy levels for the electrons.
Okay, but why do we need this energy? I'm still pretty confused.
Let me explain. The energy accumulated from the energy levels is used to pump hydrogen ions from the mitochondrial matrix into the inner membrane space. As a result, the inner membrane space has a higher hydrogen ion concentration than the membrane, creating an electrochemical gradient. This is called chemiosmosis, a process within the ETC.
Wait, so hydrogen atoms were in the matrix and now they're in the membrane? How can we get them back?
This is where it gets a little tricky. The hydrogen atoms that are now in the inner membrane space can't just cross back over because the membrane is impermeable to ion passage. They need a transporter, and that's when ATP synthase comes in.
Like an Uber driver?
Exactly. The ATP synthase moves the hydrogen ions from the inner membrane space back to the matrix, this time full of energy. These ions are accepted by oxygen, which reacts with glucose in oxidative phosphorylation. Finally, through this reaction, ADP is phosphorylated to ATP, which our bodies can utilize for energy and basic functionality.
Wow, I never even knew about the ETC and chemiosmosis. Does it only happen in mitochondria? What about like trees and spinach and stuff?
Well, I'm glad you asked.
The electron transport chain also occurs in chloroplasts through photosynthesis, the process of converting carbon dioxide, light energy, and water into glucose and oxygen.
So is that like the opposite of cellular respiration? Why is it even important?
Yes, they are opposites. Photosynthesis allows plants to use the glucose for energy and plant growth and release the oxygen into the air for us to breathe. It is crucial to life on Earth.
Okay, but what does the electron transport chain have to do with that?
The electron transport chain is a part of the light-dependent reaction portion of photosynthesis. The light energy in this process comes directly from sunlight and is captured by chlorophyll found in the thylakoid membranes of chloroplasts in plants.
So I just shine some light and get a bunch of energy?
Not exactly. In normal conditions, the photons that come from the sun travel through the thylakoid membrane until they reach the first protein complex called photosystem II. Here, energy from the sunlight is used to extract electrons from water and move them towards higher energy levels. In this process, NADP is reduced to NADPH.
Oh, this sounds a lot like the energy stuff from the mitochondrial matrix.
Yes, there are definitely some parallels. The high energy electrons then travel through the thylakoid membrane to the proton pump, where they are now at a lower energy level. The electrons are used to provide energy for the pumping of protons across the electron transport chain from the stroma into the thylakoid space.
Just like the proton pump in cellular respiration.
Very good. The energy from the electrons that is lost traveling to the proton pump is then used to pump hydrogen ions across the membrane. This produces a hydrogen ion concentration gradient in the thylakoid space, creating more energy. Finally, the energy is used to produce ATP through the previously mentioned ATP synthase. ADP is phosphorylated into ATP and oxygen is released into the environment.
One more thing. Is the electron transport chain relevant when light isn't involved in photosynthesis?
No, but we'll get there another day.
Let's not forget about the electron transport chain in prokaryotes.
Wait, there's more?
Yes, the ETC in prokaryotes is a series of proteins that transfer electrons through a membrane to create a gradient of protons. Prokaryotes generally do not contain membrane-bound organelles such as mitochondria. Therefore, glycolysis and the Calvin cycle, also vital in photosynthesis, occur in the cytoplasm.
So how are prokaryotes different from eukaryotes again?
Well, as opposed to the inner membrane of the mitochondria for eukaryotic cells, the electron transport chain for prokaryotes occur in the plasma membrane. The prokaryotic ETC has a number of electron sources and acceptors. Prokaryotes, which are chemolithotrophs, use inorganic substances as their electron sources.
And what are chemolithotrophs?
That just refers to organisms that generate energy by the oxidation of inorganic molecules for biosynthesis.
Okay, so those are the electron sources. How about acceptors?
Prokaryotes use nitrate, fumarate, sulfate, or elemental sulfur as electron acceptors. Because oxygen is not the final electron acceptor, prokaryotes must engage in anaerobic respiration.
Wow, I learned a lot. I never knew how important the electron transport chain was to our everyday lives. Whether it's in plants, humans, or bacteria, it's truly everywhere.